Hi, this video you are going to learn about cell cycle regulation. The previous video I explained what is a cell cycle. Now this is cell cycle regulation. So let's look why it is important to regulate cell cycle. Now when you look at the cell, one cell is here and the two cells after the cell cycle. That means events take place from one cell to two cells, or one cell division is called the cell cycle. So in this case, let's consider these two cells are identical. So they make exactly the same copy of the cell. Now this is important when we discuss mitosis. Mitosis is the division of the nucleus. So nucleus divide into two to make identical cells. Why we need identical cells? Now we need identical cells for many purposes. So one thing is tissue repair. So tissue is defined as collection of similar kind of cells for a specific functions. Let's say muscle tissue. These cells sometimes damaged. Then they have to replace with a similar kind of cells. Then we need the cell cycle. In this case, the word similar cells, exactly the same kind of cells we need. And you see, the regulation is just to provide the right amount of cells. Not too much, not too less. That means the speed of the cycle should be controlled. When it is required, they are going to divide. Second thing, exactly the same copy. So this is very crucial, these 2 points. Exactly the same copy, that means the sequence of DNA should be same. And the other thing, it's not too much, it's not too less, and at the right time. So all these factors come under regulation. That means you can assume cell cycle regulation is quite complex. In the basic biology, the beginners, they learn very basic steps of cell cycle. So let's look again our last lesson. We have a phase, G1 phase, S phase, G2 phase, M phase, and the cytokinases. Now, when the cell cycle undergoes all these steps, it's controlled by probably thousands of enzymes and thousands of reactions. So we have identified very important class of enzymes called cyclin dependent kinases. So abbreviation CDK. Cyclin dependent kinases are well-known set of enzymes who can regulate cell cycle. So I can draw the enzyme cyclin dependent kinases. So you will see here the active site, and also in this case they need another molecule binding on it, that is cyclin. Not only that, the activation requires the phosphorylation of the enzyme. And then this is now active complex. So I had the phosphorylation here. Now cyclin dependent kinases, upon phosphorylation, this enzyme is now active. So he is now take care of the cell cycle regulation. Now let's come into very important point, how the cell cycle should be strictly controlled. Now the S phase is the DNA replication phase. In the S phase, the DNA in a cell, if you take a human cells, it could be a 3 billion base pairs. So these 3 billion base pairs sit in an order, that's a special sequence that should be copied to the next duplicated DNA. So here semi-conservative replication, so we can have a complete replicated DNA. So this DNA, we can see the newly formed strand and still the old strand. Even though it seems very small step, that's a most important step of the cells. We know very well certain external stimuli can damage this DNA. So if DNA damage happen, let's say due to ultraviolet radiations, so this kind of mutation can happen. Then the DNA is not right DNA. So when the DNA is synthesized by the DNA polymerase enzymes, they have to copy this DNA into here. And the mutation can be happen. So during the mutation, it's not the right DNA. So all these things can be happen during DNA replications. So then we need a special system to monitor this kind of errors. So these errors should be strictly monitored. And then it's not enough to monitoring, immediately they need action plan. So we have another set of genes committed for this purpose. So well known two genes are Tumor suppressor genes and Proto Oncogenes. So this class of genes are well known for this kind of purpose, detection and the correction. So let's take one example. Let's say there's a DNA damage. So this DNA damage is detected by tumor suppressor gene, one gene called P53. So this gene sometimes referred to as guardian of genomics. So they protect the whole genomics. So this P53 gene activated upon the damage of the DNA. And then this P53 gene express to make a proteins. So these proteins known as cyclin dependent kinase inhibitor. Now cyclin dependent kinase inhibitors, they go for their mission, and cyclin dependent kinases now bind to this complex. So when they bind to this cyclin dependent kinases activated complex, they will not start, or they will not really rotate this cycle. So they can't do that one, so they have to stop. It's like a braking, it's like a controlling, it's giving the stop sign, like that kind of situations. So this is a very basic way of cell cycle regulation. So here the crucial role of cycle dependent kinases and also these tumor suppressor and proto oncogenes are important. Just to finish this lesson, in case if there is a mutation happens on tumor suppressor gene or proto oncogenes, that's a serious problem because they can't control the cell cycle. So cell cycle control is no longer their main. The cell divide. and even the DNA sequence, the newly formed cells are not correct. So just like a useless cell mass. So this kind of mass is called cancer or tumor. So let's discuss cancer and tumor in different lessons. Just to wind up the summary of the lesson, cell cycle is controlled strictly because DNA should be correct and the requirement of cells, example like a tissue repair, we need correct amount of cells at right time. This is done by cyclin-dependent kinases together with cyclin phosphorylation active complex made. These certain points called checkpoints, they regulate it. And one example, the damage to DNA, these important genes, P53 genes, activate and then make this CDKI inhibitor inhibit the complex, regulate the cycle. So this is understanding of a very basic way of cell cycle regulation. So if you like this video, please subscribe and you will get another video on next session, mitosis. Thank you very much.